let's move into bit detail of major histocompatibility complex. So let's understand MCH1 structure. They consist of two polypeptide chains, so they are called as heterodimers. Beta, 2-microglobulin chain, and alpha chain that further contains alpha, 1 domain, alpha, 2 domain, and alpha, 3 domain. This part penetrates the cell membrane. These alpha and beta chains are connected non-covalently through alpha, 3 domain, and beta, 2-microglobulin. Let's move into detailed structure of these chains. Beta, 2-microglobulin consists of 44 K-Dalton heavy chain and a 12 K-Dalton light chain, and cytotoxic T cells having T cell receptor and CD8 co-receptors. Alpha 1 and alpha 2 makes a groove to bind peptides, which is an antigen or epitope. Cytotoxic T cells also have T cell receptor, which binds with the alpha 1 and alpha 2 that will determine the antigenicity of peptide. And cytotoxic T cells have CD8 co-receptors, which binds with beta, 2-microglobulin. Let's understand MHC-II structure. It has alpha chain, which has alpha 1 domain and alpha 2 domain, and beta chain, which has beta 1 domain and beta 2 domain. This part penetrates the cell membrane. This is a groove of MHC-II. It binds much larger or longer peptides. This is structure of helper T cells having T cell receptor and CD4 co-receptors. T cell receptor binds the groove, and CD4 co-receptors bind beta 2 domain. The main difference between cytotoxic T cells and helper T cells is that the cytotoxic T cells destroy virus-infected cells and tumor cells, whereas the helper T cells secrete cytokines to activate or regulate other cells in the immune system to trigger a specific immune response. CD is a term being used in this topic. So, do you know what a CD is? Well, CD is a cluster of differentiation. It is a protocol used for identification and investigation of cell surface molecule. MHC class 1 molecule is present in all nucleated cells of the body. It means they are present in leukocytes, or WBC. They are also present in platelets, but not in red blood cells or erythrocytes of the body. MHC class 2 molecules are present in antigen-presenting cell, such as monocytes, macrophages, dendritic cell, and B cell. Let's make it very clear to you. Consider MHC as a security guard, while antigen as a thief. Let's understand the function of the MHC1 in detail. TCR, or T cell receptor, is responsible for recognizing fragments of antigen bound to major histocompatibility complex molecules. Let's understand it. When a foreign body, such as virus, enters the body, it is taken into cell. They are marked by ubiquitin. Proteasomes break this into peptide by proteolysis. The transporter associated with antigen processing, or TAP, which is essential for peptide delivery from the cytosol into the lumen of the endoplasmic reticulum, or ER. These peptides are brought to MHC-I complex through tapesin, where these peptides are loaded on major histocompatibility complex 1 molecules. Endoplasmic reticulum is a network of membranous tubules within the cytoplasm of a eukaryotic cell, continuous with the nuclear membrane. It is a network of membranes inside a cell through which proteins and other molecules move. Then this peptide and MHC-I complex moves to Golgi apparatus, then into exocytic vesicles, and then to cell surface. So this cell presents peptide or epitope or antigen to cytotoxic T cell. Cytotoxic T cell will recognize this foreign body. The infected cell will be killed by natural killer cell. MHC-I present the antigen from within the cell, so it is called as endogenous pathway. Now, let's understand function of MHC class II. But MHC-II presents the antigen from outside the cell, so it is called as exogenous pathway. The antigen is outside the cell. The cell engulfs it and forms endosome. Granules will fuse with endosome and forms phagosome. These granules has degrading enzymes that break pathogen or proteins into peptides. MHC-II are synthesized in endoplasmic reticulum. It groove temporary with invariant chain, both of which are transported to vesicle, where invariant chain is degraded. This vesicle and MHC-II fuse with phagosome and take peptide or antigen out of it. It moves to cell surface and present this antigen to T helper cell.